Would you trust research data without verification? Would you build tomorrow's medicine on unproven claims? Neither would we. In the fast-moving world of peptide science, reliable data isn't optional, it's essential. With peptides fueling advances in weight loss drugs, cancer immunotherapy, and metabolic research, the role of third-party testing has become central. It's not a bureaucratic hurdle, but the backbone of credibility, safety, and trust. What is third-party testing? Third-party testing means an independent laboratory confirms what a product really is and how it performs. Unlike in-house checks, which can be swayed by deadlines or commercial goals, external labs have no stake in the outcome. Their only job is to test, measure, and verify. These labs often hold accreditations like ISO 17025 or ISO 9001, proving their methods and results meet global standards. For peptide researchers, that matters deeply. If sequence identity or stability is uncertain, then every experiment downstream rests on shaky ground. Independent labs eliminate that uncertainty. Why independent validation matters? The real value of outside validation lies in the confidence it creates. Third-party testing removes doubt and bias, ensuring that results are built on reproducible, trustworthy foundations. It reassures not just the individual scientist, but also collaborators, funders, and regulators who need proof before moving forward. It also aligns research with international expectations. Agencies like the FDA and EMA require extensive documentation to prove identity, purity, and consistency. By relying on accredited external labs, researchers meet those standards upfront, streamlining approvals and smoothing publication or grant processes. Credibility is another advantage. Whether submitting to a top-tier journal or pitching investors, independent data adds weight. It signals that your results are verifiable, not marketing claims. And beyond credibility, there's safety. Peptides can degrade, accumulate impurities, or vary from batch to batch. Without third-party checks, these hidden flaws can derail projects. Independent labs protect researchers from wasted effort and costly setbacks. What gets tested? Independent labs run a wide battery of analyses on peptides. Identity testing confirms the amino acid sequence with methods like mass spectrometry and peptide mapping. Purity assessments then reveal how much of the material is the desired molecule versus truncated chains or byproducts. Stability studies test how well peptides hold up to light, heat, or humidity, making sure they don't break down before the work is done. Beyond structural tests, labs confirm biological function. A peptide may look perfect on paper, but still fail to activate its receptor or perform its role. Functional assays catch those flaws. Structural characterization digs deeper, examining folding, modifications like pegylation or lipidation, and aggregated forms. Safety checks for metals, solvents, or microbes provide final assurance that the peptide is clean and reliable. Together, these tests create a full picture that researchers can trust. The standards behind the science. The credibility of third-party testing comes from the standards that govern it. ISO/IEC 17025 is the benchmark for testing in calibration labs, setting strict requirements for impartiality, technical skill, and consistency. ISO 9001 adds a broader framework of quality management, ensuring continuous improvement and accountability. Beyond ISO, scientific regulators also set expectations. The International Council for Harmonization issues detailed guidelines for biologics and peptides, covering method validation and impurity profiling. The EMA recently updated its guidance on synthetic peptides, clarifying requirements for impurities, elemental contaminants, and sterilization. These frameworks ensure that testing remains relevant as science evolves, giving researchers confidence their data will stand up anywhere in the world. Current trends in peptide testing. Peptide research is growing fast. By 2023, more than 120 peptide and protein drugs were approved globally, and the list keeps expanding. Among them, GLP-1 receptor agonists dominate headlines for their role in diabetes and obesity treatments. With complex chemical modifications to extend half-life, these molecules pose new analytical challenges. Independent labs are critical for confirming modifications, spotting impurities, and verifying reproducibility. Personalized medicine also underscores the value of outside testing. In neoantigen vaccine development, labs may produce dozens of peptides for a single patient in under a month. Every batch must be validated quickly and rigorously before use in humans. The speed of production makes impartial, reliable testing more essential than ever. On a global scale, accredited testing smooths collaborations, regulatory approvals, and cross-border trade, ensuring that results are universally respected. Illuminated Peptides and Omniext Tech. At Illuminated Peptides, powered by Omniext Tech, we live by these principles. Every batch we release undergoes independent third-party testing. This is more than a formality. It's our guarantee of transparency. We submit every lot for identity, purity, stability, and functional analysis in accredited labs aligned with ISO and ICH standards. For researchers, this means confidence. It means grant reviewers trust your data, journals respect your results, and regulators accept your evidence. Most importantly, it means your science rests on verified foundations, free from doubt or inconsistency. By committing to impartial validation, we support researchers not just in generating data, but in moving discoveries forward into clinics and real-world applications. Third-party testing builds confidence, speeds discovery, and drives real breakthroughs. In peptide research, trust is everything. At Illuminated Peptides, powered by Omniext Tech, we light the path forward, trusted, transparent, illuminated, America's #1 choice for research peptides. These products are for research use only, not for human use. Like, share, and subscribe for more insights that move science forward.